Hi, everyone. I'm Chris Halcrow. I'm a research software engineer at Edinburgh, and I'm going to tell you about SpikeInterface. Now, I'm going to go through quite a lot of things that are in SpikeInterface because it can do a lot, and so I've made a little resources page that's on the Slack. And this has got links to like further documentation and kind of more information about everything that you're going to see today. So this will be like a flash highlights that's got more information. It's on the Slack, or it's here. OK, great. So hopefully, if we're here, a lot of us like eFIS data. And eFIS data comes in lots of different shapes and sizes. There's tetrode data, there's silicon probes like NeuroPixels and Cambridge Neurotech, and then there's higher density probes, including these in vitro ones that are like 4,000 channels sampling all at once. So hopefully we all like eFIS data. But unfortunately, to use it, we have to do spike sorting, which is horrible. So spike sorting is the process of detecting spikes from your eFIS data, and then clustering those spikes into units, and those units are meant to represent single neurons. And you get out like each single neuron and the spike times. OK, great. But there are many problems. So maybe in your lab, you have some pipeline. You read in a SpikeGLX file, you do kilosort 4, and you export to Phi. But there is not just SpikeGLX. There's like 20 different recording formats you have to deal with. There's not just kilosort 4. There's lots and lots of different spike sorters. And even kilosort 4, there's many like little variations in its versions as it's released, as with all of them. And it's not just Phi you can export to. You can export to loads of different formats. And there's a basic problem, which is just, how do you open these? Even opening them is difficult. Once you've opened them, how do you visualize them? How do you compare them? How do you run all these different sorters? How do you put everything on a level playing field? And hey, the postdoc who did this recording and did this sorting left the lab 3 years ago. Can you possibly open their data? So that's a terrible problem. We're all very sad. But I've got good news. And that good news is SpikeInterface. So the idea of SpikeInterface is to kind of abstract away all those complications and annoying things about formats. So we hope to try and read in any raw data format. We hope to kind of wrap as many spike sorters as possible. We take this raw format, then we kind of wrangle it into a standardized form, which we can then feed into any of these different spike sorters. That exports like a sorting object, and then you can export that to any of those sorting formats, and you can also read in those sorting formats if you find them on some hard drive that's lying around the lab somewhere. And then, once you have your recording and sorting, we've also introduced a kind of post-processing object called a sorting analyzer. And that contains all the things you might be interested in, waveforms and spike amplitudes and that kind of thing. So the issues that we're trying to solve is these incompatibilities between all these different file formats. We're trying to solve that it's quite hard to run spike sorters if you want to try and run all 10 of them. So we try and make that easy. And also, we're pretty careful about provenance. So that means if you make a sorting analyzer, you export a folder, all the information is there in the folder to be able to recreate that from scratch, given the initial raw data. So we're working hard to include all that. OK, that's the kind of overview. So now you'd usually go through a pipeline, and I'd show you how it works. And at the end, we would get out this nice sorting analyzer object. Instead of doing that, I'm going to show you the sorting analyzer to begin with to try and convince you that it's worth putting your time in investing in this to make one. So as I said, the sorting analyzer is a kind of post-processing thing that combines the recording and the sorting, and you compute extensions like waveforms and quality metrics and parallelograms and things. And the idea for us is this is a kind of pretty standard format that can be used to build other tools on top of. So I think I'll just show you one. Anyone know how to zoom in on Finder? Zoom. OK, doesn't matter. Here's an analyzer. It contains like information about the recording. It contains information about the sorting that you've done. And then it has a bunch of these extensions, which are really just simple like MPZ files and some information about how they are computed. But once you have this, you can run tools such as SpikeInterface GUI, which is a way of looking at your results. So this is the kind of information it contains. This GUI is a very fast thing to look through units, and you see lots of information about them, their amplitudes and amplitude distribution, their parallelograms, their waveforms. I really like the waveforms, so let's close some of these. And then we can quickly flick through and have a look at what looks like a good single unit, what looks like noise, and so on. So once you have a sorting analyzer, this tool is available for you. There's a web version as well. Also, you can just load it in to a Python script or a Jupyter notebook. And once you've loaded it, you could do a similar thing than we just saw in the GUI, but actually make a figure out of it and look at your spikes, look at your units. And you can extract things like spike trains easily. Also, you can do cool things like this. So this is taking the sorting analyzer. There's a bunch of quality metrics that have been computed, and there's a trained model which is now going to curate for us whether units are noise or good units, which has been trained on some manually curated data. So this model has told us that unit 3 is definitely noise, but unit 372 is good. So let's have a look at unit 3. Yeah, no spatial decay, terrible parallelogram, looks like trash. Unit 372, that's nicer. Cool. So as soon as you've got a sorting analyzer, you can apply these models. Yes? Mm-hmm. Yeah, there could be different reasons for why the amplitude changes with time. So the amplitude is like the amplitude on the channel that has like the maximum amplitude on it. So if your probe is moving a little bit, that can affect the amplitude size, for instance. But there might be other reasons.
I mean, the fact that the amplitude seems to be varying like smoothly suggests that a single unit time, that's like the shortest amplitude.
Yep. And we try and not take a side about this. We try and say, okay, we give you the tools to look at this, and you decide what's going on. So have fun. And also, other tools interact with SpikeInterface. So for instance, NeuroConv has a function which is take a sorting analyzer and spit out an NWB file. This probably isn't the recommended way to make NWB files, to be honest, but this works. It spits out an NWB file that contains the recording and the sorting information, and because those are tied together, you're kind of sure that it's working like you're expecting. So I hope now you really want to make a sorting analyzer. There's other tools as well, things like UnitMatch to do matching between days, things like Lusac, which does comparisons of many different sorters. They start from sorting analyzers. Okay, so now you want to make an analyzer. So now your question is, but Chris, how can I make a sorting analyzer? Well, good news, it's very easy. So here's a very basic pipeline. You import the SpikeInterface package. You read your OpenEFIS file. You apply some pre-processing. You run kilosort. You make one of these sorting analyzers. You compute some extensions. Okay, what if I want to run mountain sort instead of kilosort? One thing's changed, no problem. Oh, my lab has changed from OpenEFIS to SpikeGLX. Hey, no worries. I'd like to also compute the spike locations. Add it into the extensions. I forgot to add a bandpass filter. That's okay, one more line. I've done a multi-shank sorting, and I want to sort by shank rather than everything all at once. You do a split by, everything works the same in like a week's time. There's my one lie of the talk. How about if I've got 2 recordings, I want to concatenate them and then sort them. You just read them both in, you concatenate them, everything remains the same. And I want to do some motion correction. Well, that's just another preprocessing step. I'll get into some of this in more detail later. But the point of this is, with SpikeInterface, spike sorting should be easy, and it should be easy with any file format you have, any preprocessing steps you want to do, and any spike sorter you want to run. And in the end, it spits out a standardized object that you can share between labs. We're trying hard to make sure that this object will like survive across time, that you can open it in 10 years' time. And also there's a bunch of like comparison and visualization tools built on top of it. It sounds like we're trying to, I don't know, compete with the NWB format or something, but it's not that. At the base of it is all like similar tools and similar people working on these things. We're definitely working together, not like in opposition to each other. Okay, if you want to just get started with SpikeInterface, there's an Introduction to SpikeInterface course that goes from like the very start. It goes from like making virtual environments in Python, and by the end you have a pipeline that will work in your lab. So it's like a 0 to hero course. It's like 6 10-minute videos, takes you through the whole process. And that's on the resources page as well. Okay, that's the kind of like broad overview stuff. So any questions at this point? Super. Yep. I'll talk about that a bit more later, but basically there's any models. Also, you get a sticker. But yeah, I'll talk about that later. Okay, let's now kind of specialize into what this conference is about and think about SpikeInterface, Neurodata Without Borders, and Dandy. Okay, I'm going to try and do something brave and install SpikeInterface from scratch because I really like a new Python packagement system called UV, and I just want to show it off because I no longer think about virtual environments, and it's great. And also, for instance, hospitals in the US can no longer use Conda because of like a license. Well, some hospitals, Harvard Medical School, no longer use Conda because of some like licensing paying issues. We have to move away from Conda. So my suggestion is UV. Okay, I'm in my projects folder. I'm going to make a directory not called talk demo, that already exists, called talk demo 2. That's a joke. Okay, I go into it. I'm using a tool called UV. I'm telling the folder this is going to be a UV folder. UV is going to manage a Python environment in here. I'm going to add the following packages. This is like pip install. SpikeInterface, Dandy, SciPy, IPyKernel, Matplotlib, blah, blah, blah. That's built my environment right there and then. And then I'm going to open VS Code. I'm going to run VS Code telling it that I'm using UV. So VS Code knows about the environment that I'm opening it with. And this has, on the fly, made a virtual environment when I open VS Code. So every time you run Python, it creates a new virtual environment and keeps all your packages up to date. So if anybody wants to talk about Python packaging, please come and talk to me. I love this stuff. Okay, now it's not going to work or something. Yes? Yep. I'm always keeping up to date because I'm like developing. So I like to keep things like as up to date as possible. But you can like freeze packages, their versions where they are. And so you can say, oh, I'm using Kilosort 4.0.23 or something. That's easy to do. You just, when you add it, you can do like equals, blah, blah, blah. Yep. Very similar to pip install. And the nice thing is I now log on to my supercomputer. I run that command and I've got the same command. I didn't have to do any syncing. I just have that one thing. So we use this on like 4 different compute systems in Edinburgh. And this is the way we do all our Python things. UV can also install Python versions. So if I want to do this same thing, say if I want to open VS Code, but I know for some reason I want to use Python 3.8, no, not 3.8, 3.10, I can do this and it will open it with a different Python version. Whereas with virtual environments, you start with a Python version and make them. So UV is really great, everyone. Okay. And if you run that, you will create the same environment that I just made. So I just installed SpikeInterface and all the dandy API stuff in a oneer. Very easy. If you're following along, you can create that environment right now. Okay, but this talks about SpikeInterface. So it'll be the only environment that's around. Yes, thank you. Okay, I'm now going to get a data set, thanks to Stephanie's advice in the talk. I'm doing something where I get the dandy ID, I get the assets from the ID, and then I don't have to copy any file names and stuff, because that was annoying. Okay, so we have an S3 URL. And SpikeInterface has a function called read NWB recording. And if you give it the S3 URL from Dandy and you tell it to stream, it will try and read it straight from Dandy, and it won't download it. It's just streaming it from the interwebs. An error. Oh, no. The error says there are multiple electrical series found in the file. Please specify the electrical series path. And that's because there's a LF band and an AP band. For this, let's get the AP band. Path or name? Path. High wire stuff. Okay, so as I said, the kind of first important object in SpikeInterface is the recording. So what's happened is we've looked at this dandy set. We've downloaded all the metadata, so we know about the recording, but we've not downloaded any data. We'll only download the raw data when we ask for it. So this recording is 384 channels, 41 minutes. It's in 16. You can look at channel IDs. You can look at things like, is it grouped in any way? Blah, blah, blah. And we can also do things like plot the probe map. I'm just going to do a widget command as well. Whoop. And what comes out is a very ugly probe, but this contains all the electrodes and their XY positions relative to the probe. So you can start visualizing information quickly in SpikeInterface, which is nice. Okay, now let's look at some raw data. Okay, what I'm going to do is plot traces, and plot traces is going to request the actual raw data. So this will take 10 seconds or something. That time is just it streaming the data from Dandy. Now, actually looking at raw data is a bit pointless without any pre-processing. It looks like some lines. This is the electrical series over time. So we've got to do some pre-processing. So what we can do is just apply some pre-processing to our recording. So this is me doing a bandpass filter to the recording, and then doing a common reference median filtering to the recording. And now, because SpikeInterface is also based on this kind of lazy, you only do things when you ask for it thing, we're streaming the data from Dandy, and then when we get it, SpikeInterface applies these pre-processing steps only to that little chunk of data that we've asked for. And I forgot to uncomment that out, which is fine. Okay, so everything's lazy, so this streaming method really works nicely with SpikeInterface. And these maps are,
You're loading 1 second at a time?
Yes, this is 1 second at a time. There's a window that you can adjust here to see more. Then if you zoom in on these things, okay, that might be a spike. So what these colors represent, this is time, this is 0.004 seconds. Noise, 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 noise. Electrical signal gets negative, and then positive, and then back to noise. That's a spike. So we've streamed and pre-processed the raw recording live, and we're visualizing a real spike in the data. So Pretty cool. Okay, great. Also, as well as looking at raw data, instead of doing read NWB recording, we can do read NWB sorting. And then we get a sorting object out, which has unit IDs and things, and we can do something like plot a raster map of the units. So SpikeInterface just gives another way to interact with data on Dandy. It's using all the same like technology in the backend, but this is nice because now we can actually go and like pick out the spike trains that we want, and we can answer some computational questions. Cool. One thing I was going to say is, right, we've all now started looking at Dandy sets, and probably you're finding it can be quite difficult to know what's going on with them and to like reproduce anything from them. And we in our lab, this is kind of like a poor man's version of what Carter is doing. But for instance, we have a README page about our data and how it's organized, and then we've got little code snippets that do things like, actually the best one is a pineapple one. We have code, for instance, that just makes a position tuning curve from the data. And I really think going forward, if you want your data set to be used, it's good just to make a little website that you can recreate simple things in it using. And just using GitHub Pages is fine. So if we see this example code section, actually writing this in code is incredibly simple. This is very easy to set up this type of website. And if anybody wants to like make this type of simple README page, get in touch. I think it's really important for putting your data online. I think it's not enough just to upload it onto Dandy. Or use maybe the AI tools will be able to do this without us putting effort in, which would be good. Okay, great. So that was the SpikeInterface plus NWB plus Dandy bit. Any questions? Yeah.
So you're going to use SpikeInterface and use iSort to separately upload those
Yeah, no, so we put it on the user to install, but all the installations almost, okay, I'll tell you a different thing in a second, but it is as that is. It's usually as simple as a pip install. Nope, okay. It should be as simple as a pip install. But I'll tell you a different way to do it to bypass the sorting, to bypass the installation in a second. No, I'll tell you that immediately. Okay. One cool thing about it is that we also containerize things. So if you can get like Docker or Singularity installed and working, then we provide, the way to think about these things is like little applications with an environment already built inside of them. And that lives somewhere. And you access that through Docker. And provided Docker is working, which is non-trivial slightly, then you can get an environment with all the packages pre-installed. And this also includes the MATLAB sorters. And one cool thing about that is that MATLAB costs money, but actually it's legal to use the code in a container even if you don't have a license for MATLAB. So if you can get this working, you don't need to install MATLAB or have a MATLAB license to be able to run the MATLAB sorters. So if you really hate installing Python packages and MATLAB packages, you can switch to hating installing Docker instead. So that's meant to be the solution for that. It works pretty well. My experience is it works fine on Linux, and it's pretty grim on the other OSs.
Yeah, how easy would it be to like configure some custom sorter, some executables?
Yeah, very easy. So I won't show you anything, but there's a module in SpikeInterface called sorting components, and it tries to like break sorting into its individual components. So most of them have some like spike detection method to do first. And then once you've detected your spikes, it maybe builds a template library or it does something else. And the idea of that module is it splits it into like 8 steps or something, and then you can configure that very, very finely based on your data. But that's quite like an advanced specialist thing to do.
I mean, if I have like an executable
Oh,
that is not a known executable, like I don't know, one of those popular spike sorters, I have an executable. How easy is it to like interface that?
yeah, I imagine not very. Stickers. There's more stickers. Okay, great. Yeah, thanks for the question. Yes. Mm-hmm. Yep. All good. Yep. So we run, in our lab, we run Kilso 4 on a GPU through SpikeInterface. Then it'll run on your CPU. And Kilso 4 is pretty fast on a CPU. By fast, I mean like an 8-core CPU will do an hour Kilosort 4 NeuroPixel in 8 hours. That's overnight. That's pretty fast. And CPUs are cheap. I think it's actually more cost-effective to run Kilosort 4 on a CPU than a GPU if you're doing like Amazon cloud services. Okay, now I'll just show you some fun bits and pieces. Oh, I showed you this visualization, so you've seen that. Another thing is eFIS files are big. You can compress them using the ZAR format and, I forget, the wave pack compressor, which is some lossless audio compression. And you can reduce your NeuroPixel 2 recording down to 40% of its original size. All you do is load in your recording and then save it again. And if you go to NWB, NWB kind of requires compression. So it's the default. There's some compression default in the NWB format when you make an NWB file as well. Or at least.
I believe we're doing compression by default in Neurocom.
Yep. If you put an NWB file through the Dandy checker and it's not compressed, then it complains. So I think everything on Dandy is compressed, right? Yeah, okay. So you can compress things, so that's good. Motion correction is an issue for acute recordings. So this is lots of spike locations, and this probe has been deliberately wiggled. And you can see that the spike locations change. This is a problem for spike sorters because the template of the spike that you get is different here or if it's there. And this breaks a lot of the assumptions in the algorithms. So people have tried to solve this by applying motion correction as a pre-processing step. There's quite a lot of different algorithms, Dredge, Medicine, the algorithm from Kilosort stripped out, those kinds of things. We allow you to run those in this correct motion function, and it also gives some output. So you can see, like here on the left, these are spike locations before motion correction and after. So you also get some visual output to see if your motion correction was successful. So that's nice. We've just added something to share pre-processing pipelines between labs. So I was talking a lot about the sorting analyzer. If I go and ask Alessio at Allen for one of his sorting analyzers, I can take that, apply this, get a pre-processing dictionary from his analyzer, and I don't have to trust him about what he did when he was pre-processing his recordings. I'll get a dictionary that tells me exactly what he did, and then I can apply that to my recording in my lab. So instead of having to go through the whole process, I can just say, hey, send me an analyzer or give me this dictionary that describes the entire pre-processing process. So this should make sharing different pipelines between labs very simple. I spoke about containers. Oh yeah, I was going to talk a little bit more about this automated curation. So once you have a sorting analyzer, SpikeInterface has implemented about 40 different quality metrics. Simple ones are like signal to noise ratio or ISI violations. And you can use these to curate your units. So I say, hey, I'm only going to include units with a high signal to noise ratio and a low number of ISI violations. So you can do basic thresholding. Or there is a recent paper where roughly some people from the Musel lab curated about 3,000 NeuralPixel 2 units into like noise, SUA, or MUA, and then trained a model based on this curation. And you can just try out that model on your analyzer. And the nice thing is it's up on the Hugging Face hub, which is a repo of like machine learning models and stuff. So you can simply give it your analyzer, give it this repo ID, and it'll spit out these labels that it thinks, whether your units are good or bad and things. So there'll be more models on this soon. Another workflow you might have is, okay, I've got 100 recordings. I'm going to manually curate 4 of them. I'm going to train a model based on that curation, and then I'll apply that model to the rest of my recordings. So that should significantly reduce your curation time. And you can do all that within SpikeInterface. You just give it the analyzers and the labels. It'll train up a model. Then you can take this model and apply it to other analyzers using this function. So very fun. I'm not going to talk about sorting view because Jeremy's here. He might talk about it. And yeah, so that's some highlights. There's now quite a lot of people who work on SpikeInterface. Alessio and Sam are the lead developers. We all have like some of our time working on it or just voluntarily working on it. And now we're approaching 100 people who have contributed to SpikeInterface on the repo. When we're applying for funding, funders genuinely ask how many GitHub stars you have. So if you use SpikeInterface or like SpikeInterface, we'd really appreciate it if you starred the repo, because it's a genuine metric nowadays. So sorry to ask, but thank you. Okay, great. That's my talk. Cool, I think we're good. There was lots of questions during, yes. The GUI. Right, you would like to do merging and splitting. Whoops. Right, so what we can do, oh goodness. Okay, these tools exist. No, the merging exists. Let's see if we can find 2 units that are similar. What we can do, there's a way to sort. Yeah. So for instance, it's very, okay, just curating, doing like, right now I'm labeling things, noise, good. That's all labeled now. The merging, there's a way where you sort by template similarity, and then you can view the 2 waveforms at once next to each other. Now the way you do that is simple, but I can't remember. Oh, merge list? Oh no, that's doing automated merging. Yeah, I've not actually worked on this at all, but it is simple. And then splits, there is a PR at the moment for implementing splits. So hopefully in the next couple of weeks. And the way those will work is on like your amplitude plot, you'll be able to like select things and then either say disregard those from the unit because I think that they're too far away, or split that into another unit. But yeah, all that stuff is in the works. Yeah. Yeah. So this is just like a one sorting at a time system. The tool that's been developed that I would use is this LUSAC by Aurelian W. And they do they have things like they use some graph theory methods and try and link units from different sorters that look similar. And you do it with many sorters, and then different ways that the graph can combine suggests this is a merge or this is a split. And then it does a consensus sorting based on that kind of thing. It's a little bit of work to get going, but it's very cool once it's going. But for doing multiple sorter, okay, yeah, this is one at a time. Yeah.
When I used OpenInterface,
Yep.
my raw data is not just including this channel, but also there's some analog signal that is also. So my OpenInterface raw data is not just about spiking information, but also has some behavioral, some output data from the analog. And I wonder how this spike interface can determine which signal to take it for sorting. And maybe in the future, there's a way to align it and using those other signals to view or visualize my spiking activities.
So is it saved on an ADC channel in the...
Exactly, yeah.
So I think, wait, is it saved separately or is it saved in the same?
It's in the same, it has a row together.
So I think this depends a little bit. So when you do a read like this, you specify which series you're looking at, and it will often default to look at the raw electrical series. But if you change this, actually, this isn't really the right function. If it was read OpenInterface, actually, let's go look at it. It has a stream ID and a stream name, and specifying different stream names should load in different data. So for instance, the one I know about is that we save TTL sync pulses, and that you can read in as a different stream like that. And it still looks like this recording object. It has all the time information in it and all that stuff. So hopefully easy. Ben?
What's the workflow currently you would recommend for if you had longitudinal recording and you wanted to? What's the workflow you would recommend currently if you had a longitudinal recording and you wanted to?
Sorry, what is longitudinal?
Meaning you had multiple sessions that potentially were recording from the same neurons.
Yep.
And you wanted to see if you could match them across sessions.
Yeah. How much? What's the time difference between the recordings?
I don't know. It's more of a hypothetical question.
Yep.
I think I talked to a lot of groups that are interested in this kind of thing. Spanning from, 2 sessions in the same day to up to a month.
Yep. So, right, there's 2 basic approaches. And one is either concatenating the 2 recordings and doing the sorting all at once on them. And then the hope is that if you do some motion correction, if there's some time in between, that will kind of allow it to look a bit more continuous for the sorter. And then the nice thing about that is that you have a consistent unit ID identification throughout the whole thing. You'll probably get a bunch of splits and that kind of thing. Then you need some sort of tool to maybe do some merging afterwards, like SpikeInterface GUI. The alternative is to sort each session independently and then try to do unit matching afterwards. And unit match is pretty good for doing that, but there's not really been a study to say which one is better. In my experience, it gave pretty similar results, actually, but that was looking at sessions that were on the same day, but an hour apart. And that was also chronic, which will change things. But yeah, there's those two workflows. I don't know what to recommend, but they're both very easy to implement in SpikeInterface. Yeah. You have to apply that grouping. A few different things, I think. One is like the grouping information is stored differently depending on the format. So for instance, if you're doing like a bundle of tetrodes, you have to kind of specify the grouping yourself. So we probably want the user to be thinking about the grouping, I would say. So, and you might not necessarily want, in the tetrode case, for instance, you probably want to do some of your pre-processing on everything. If you do like a common median on one tetrode, it just wipes out the signal. So it's all a bit subtle, and it's worth thinking about from the user. So we probably want them to think about it. Yeah, and you can even do like some pre-processing on the full thing, and then some extra pre-processing on the grouped stuff, and then sort it by group, or reconcatenate it and sort it all together, which might be appropriate in some. Yeah, that's up to you. If you just kind of followed the tutorials, you'd probably end up with one analyzer, but then the units are sorted by group. So it's quite easy just to pick out like group 0 and stuff. But it'd be pretty easy to make a dict. You just need to add a for loop somewhere. Okay, great. Our lab's pipeline is SpikeInterface followed by Pineapple. So I'm very happy that this talk is SpikeInterface followed by Pineapple. So yeah, let's end it there.